everyone. In today's session, we'll explore the remarkable metabolism of the erythrocyte. And this is a cell that carries oxygen for the entire body, yet lacks a nucleus and the mitochondria. And so we're going to see how does it produce energy without a mitochondria. Look at how it defends itself against oxidative stress and regulate oxygen delivery to tissues. And so by the end of the session, we're going to see how the erythrocytes generate ATP via glycolysis, how the pentose phosphate pathway equips the cell with NADPH, which is required to combat oxidative damage, and also the Rapaport Luebering Shunt, which fine-tunes hemoglobin's oxygen affinity. And so we'll be able to appreciate how these interlinked pathways allow erythrocytes to survive and function. The main source of fuel for erythrocytes is glucose, and the bulk of this glucose enters glycolysis and it is broken down into pyruvate and lactate. And so let us look at glycolysis step by step. When glucose enters the cell, it is immediately trapped by phosphorylation as hexokinase converts it into glucose 6-phosphate using 1 ATP. Glucose 6-phosphate is then rearranged by phosphoglucose isomerase into fructose 6-phosphate, which is further phosphorylated by phosphofructose kinase 1, the committed and highly regulated step of glycolysis, to form fructose 1,6-biphosphate. Aldolase will then split fructose 1,6-biphosphate into two 3-carbon molecules, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. A triphosphate isomerase quickly interconverts dihydroxyacetone phosphate into another glyceraldehyde 3-phosphate, ensuring both molecules can continue down the pathway. Each glyceraldehyde 3-phosphate is then oxidized by glyceraldehyde 3-phosphate dehydrogenase, producing NADPH and forming 1,3-bifosfoglycerate, a high-energy intermediate. This molecule then donates a phosphate group to ADP via phosphoglycerate kinase, generating ATP and yielding 3-phosphoglycerate. Through the actions of phosphoglycerate kinase, 3-phosphoglycerate is rearranged and converted to 2-phosphoglycerate, which is then dehydrated by enolase to form phosphoenol pyruvate, which is also another energy intermediate compound. We then have pyruvate kinase, which catalyzes the transfer of a phosphate group from phosphoenol pyruvate to ADP, generating ATP and pyruvate. Pyruvate is then converted to lactate by the action of lactate dehydrogenase, producing NAD+, which is then reused in glycolysis. So having seen how glycolysis takes place in the erythrocytes, We then ask ourselves, how is ATP utilized in these cells? And you see that ATP powers the sodium-potassium pump, keeping sodium low and potassium high inside the RBCs. And this is crucial for preventing osmotic balance and flaring or lysis. ATP stabilizes interactions between cytoskeleton proteins and the plasma membrane, and this keeps the RBCs by concave shape and flexibility so that it is able to squeeze through narrow capillaries. Further, we see that ATP is also used to pump calcium out of the cells, thereby maintaining very low intracellular calcium ions, and this will prevent unwanted activation of proteases and also membrane rigidity. In phosphorylation of membrane proteins, we see that ATP is required for kinase, that is kinases that phosphorylate pectin and other cytoskeletal proteins, and this helps regulate erythrocyte deformability and membrane stability. And finally, though an indirect role, ATP fuel the metabolic processes that keep hemoglobin in a functional state and prevent premature hemolysis. In red blood cells, glycolysis is not only about generating ATP, but it also serves a special role in regulating oxygen delivery. This is where Rapaport Luebering Shunt comes in. Normally, as we've seen, glyceraldehyde triphosphate is converted to 1,3-phosphoglycerate, which is then converted to 3-phosphoglycerate by phosphoglycerate kinase, and this produces 1 ATP in the process. However, there is a clever detour. Through the actions of biphosphoglycerate mutase, 1,3-phosphoglycerate is converted to 2,3-biphosphoglycerate, a unique compound in the erythrocyte. And this compound binds to deoxyhemoglobin and stabilizes it, lowering hemoglobin affinity for oxygen. And this means that hemoglobin will release oxygen more easily to tissues, especially when it is needed most in hypoxic condition, at high altitude, or during anemia. And so without 2,3-phosphoglycerate, hemoglobin will hold on to oxygen too tightly and tissues would be deprived it. So we are saying that 2,3-phosphoglycerate fine-tunes oxygen delivery by making sure hemoglobin lets go of oxygen when the body needs it. Once 2,3-phosphoglycerate has done its job, it does not accumulate indefinitely. An enzyme known as 2,3-phosphoglycerate phosphatase will convert 2,3-phosphoglycerate into 3-phosphoglycerate, which re-enters glycolysis further downstream. However, it is important to note that this shunt bypasses the ATP-producing step at phosphoglycerate kinase, and this means that every time erythrocytes make 2,3-phosphoglycerate, they sacrifice 1 molecule of ATP. The other fate of glucose in the erythrocyte is the Pentose Phosphate Pathway, also known as the hexamonophosphate pathway. And the pathway unfolds in two phases. We have the oxidative phase, which generates NADPH and ribulose 5-phosphate, and the non-oxidative phase, which shuffles carboskeletons to provide flexibility. So let's look at the oxidative phase. And here we have glucose 6-phosphate converted to 6-phosphogluconol delta lactone through the actions of glucose 6-phosphate dehydrogenase, generating one molecule of NADPH. Further, the lactone ring is hydrolyzed into 6-phosphogluconate, and this is a simple housekeeping step catalyzed by lactonase necessary to keep the pathway flowing. 6-phosphogluconate is further oxidized and decarboxylated to release ribulose 5-phosphate, and another molecule of NADPH is produced. And so at this point, the cell has already earned 2 molecules of NADPH per glucose 6-phosphate. In the non-oxidative phase, the ribulose 5-phosphate can undergo isomerization, generating ribose 5-phosphate through the actions of ribulose 5-phosphate isomerase, can also undergo epimerization to form the loose 5-phosphate. Through the action of transcatolase, which requires thiamine as a cofactor, 2 carbons from the loose 5-phosphate are transferred to ribose 5-phosphate, generating glyceraldehyde triphosphate and pseudoheptulose 7-phosphate. A transaldolase transfers 3 carbons from pseudoheptulose 7-phosphate to glyceraldehyde triphosphate, generating fructose 6-phosphate and erythrose 4-phosphate. Another transcatolase reaction transfer 2 carbons from a second cellulose 5-phosphate to erythrose 4-phosphate, forming another fructose 6-phosphate and glyceraldehyde triphosphate, which are intermediates that can re-enter glycolysis. So one may ask, why is the pentose phosphate pathway important? We see that for tissues such as liver or the rapidly dividing cells, the pentose phosphate pathway is important for both NADPH and ribose 5-phosphate production. However, in the RBCs, the pathway's main mission is generation of NADPH, which keeps glutathione reduced. And reduced form of glutathione neutralizes hydrogen peroxide and reactive oxygen species that constantly threaten hemoglobin and the cell membrane. If this pathway is impaired, for instance, in a deficiency of glucose 6-phosphate dehydrogenase, the erythrocytes become vulnerable to oxidative stress, which may arise from infections or from certain drugs, and this can lead to hemolysis and anemia. Thus, the pentose phosphate pathway is less about energy and more about survival. It equips the RBCs with the biochemical armor it needs to endure its long 120-day journey through the bloodstream, constantly exposed to oxygen, yet never destroyed by it. The other pathway seen in the erythrocyte is a methemoglobin reduction pathway. And this pathway keeps iron in check. Remember that hemoglobin's ion must remain in the ferrous state to be able to bind and carry oxygen. However, iron tends to oxidize to the ferric state, forming methemoglobin, which cannot bind and carry oxygen. And so to fix this, the erythrocytes have the methemoglobin reductase pathway, which utilizes NADPH from the Pentose Phosphate Pathway and NADH from Glycolysis to activate the functions of NADPH methemoglobin reductase, which reduces ferrous ion, which reduces ferric ions back to the normal state, that is a ferrous ion. And so this ensures that hemoglobin is maintained in an oxygen-carrying form and ensures continuous and reliable oxygen delivery to tissues. Other pathways related to erythrocyte is the purine salvage pathways, and this is very important because erythrocytes cannot synthesize nucleotides since they lack nucleus, as earlier said. And so they recycle the purine bases to maintain ATP and GTP pools. So in summary, this is what we can say, that the erythrocyte, despite lacking nucleus and mitochondria, they maintain a delicate balance. Glycolysis fuels its energy needs. The pentose phosphate pathway shields it from oxidative harm, and the Rapaport Luebering Shunt fine-tunes oxygen delivery, and reduction pathways keep hemoglobin function. And so together, these interconnected metabolism streams form a silent processes or a process that enables the RBCs to function without a nucleus and a mitochondria. And so with that, we come to the end of this session where we have looked at the 4 major processes, metabolic processes that occur in the RBC.